CD20 is a transmembrane protein found on the surface of B lymphocytes, which is a white blood cell responsible for the production of antibodies. CD20 is a key target for B-cell-mediated diseases, and lab-produced monoclonal antibodies can be used as a therapy for such diseases. The exact mechanism of action of monoclonal antibodies acting on CD20 is not completely elucidated. However, they do exert powerful cytotoxic effects on B cells. The mechanisms of action in which monoclonal antibodies are thought to kill B cells is through direct initiation of apoptosis, antibody-dependent cell-mediated cytotoxicity, and complement-dependent lysis. CD20 is often targeted in cases of B-cell dysfunction, which occur in leukemia, non-Hodgkin's lymphoma, and multiple sclerosis, which is the focus of our designer drug. It is worth noting that links between CD20 and diabetes have been postulated but have yet to be fully described, and trials for rheumatoid arthritis are also ongoing. Multiple sclerosis occurs when the central nervous system becomes inflamed and causes myelin sheath and axon damage. Traditionally T-cell mediated, B cells still contribute to this by producing cytokines which drive an inflammatory response.
There is ample evidence to suggest that Abermumab is a well-tolerated medication, with little to no safety risks identified. Abermumab is a fully human antibody, therefore it's less immunogenic than other monoclonal antibodies, such as rituximab. Despite this favorable profile, Abermumab is not perfect. The principal issue is that it cannot cross the blood-brain barrier to elicit central effects. There are numerous problems tied to this. Some methods which enable drugs to cross the blood-brain barrier include reducing its size, increasing lipophilicity, or reducing polarity. However, alterations such as reducing Abermumab's size may diminish its potency. Additionally, increasing lipophilicity by addition of a lipid coat would only enlarge the molecule even further. And finally, methods for reducing biologics polarity are limited.
To bypass the above issues, scientists from Roche developed a brain shuttle that utilizes the receptor-mediated transcytosis of transferrin receptors. Transferrin receptors found on the walls of brain capillaries mediate the transport of transferrin protein from blood into the extracellular space of the brain. Attaching a single-chain Fab fragment of an anti-transferrin receptor monoclonal antibody to the Fc region of an anti-CD20 monoclonal antibody, the complex can penetrate the vascular endothelium into the brain extracellular space, alongside the original cargo of the transferrin receptor. In this way, the engineered antibody can bind to its target on autoreactive B cells, which in the brain contribute to the progression of neural dysfunction in multiple sclerosis. However, this sudden increased pro-inflammatory response associated mainly with the complement cytotoxic activity mediated by the monoclonal antibody could be devastating for the nearby healthy neuron cells, resulting in numerous adverse effects.
By glycoengineering the sugar structures of the antibody Fc region, this issue could be resolved. It was previously shown that blockade of fucose attachment to the core of the asparagine 297 associated sugar chain by the addition of the arabinose, a fucose analog, to core N-acetylglucosamine decreases the complement-dependent cytotoxicity and instead relies more on the antibody-dependent cell-mediated cytotoxicity and phagocytosis. This would decrease the potential damage to healthy cells. Additionally, even though the monoclonal antibody is fully human, it is produced in murine myeloma cell line NS0. The majority of mammals, mice included, express an enzyme that is inactive in humans, CMAH. It hydroxylates the terminal sialic acid N-acetylneurominic acid to produce N-glycolineurominic acid. As we are exposed to the N-glycolineurominic acid molecule due to ingestion of animal produce, humans have developed antibodies against it. To further prevent immunogenicity related to the monoclonal antibody administration, the CMAH enzyme in the murine cell line could be knocked out, inhibiting the synthesis of N-glycolineurominic acid, resulting in the expression of N-acetylneurominic acid form, which not only decreases the dangerous pro-inflammatory complement-mediated activity of the antibody, but also minimizes the risk of antibody elimination from the circulation.